Hello, Jesse. Yeah, we did have a DevoWorm last week. It's recorded. It was a pretty nice set of updates on GSOC too. So hope to go back to that. Hello.
Hi.
Hi, how are you?
Oh, I'm still getting data.
Okay.
So it sort of works.
Well, this is for your tensegrity stuff?
Yes, I've got my 3 models and actually 4 models because they made the posts a different size and they seem to behave differently with that. So yeah, but I have to get each data point separately, so it's being sort of longer process, and yeah.
Well, that's good. Sounds good.
Well, it's all I can do, and hopefully I'll get it all done in a couple of weeks before the CompuCell 3D course.
Oh, yeah. That's coming up.
Yeah. So I'm supposed to check that out so I know how to work with it. Yeah. Anyway.
Yeah.
I'm just model crunching or something.
Yeah, model crunching. That's a good word. All right. Well, yeah, thanks for the update. So, Mehul, how are you?
Hi Bradley, hi everyone. I'm good. Thanks for asking. So there is a small blackout here. That's why I can't turn on my camera. It's completely black.
Okay.
Otherwise, I wanted to share something. There's a small update.
Okay.
So based on the last week's discussion, we have explored more static graphs, spectral, but we also we do with the lineage hypergraphs. So based on discussions and outcomes, we propose the dynamic schedule. So that is continuous time exercise from 1 to 90. So what we can try creating time point. So based on the distance threshold of 5, we have created the hypergraphs for every time point, and that is how it looks like. So because it was taking too much time to create hypergraphs for all 90 time points, so this is a time point like this is 1, 11, and 21, and as you can see, it is getting lesser and lesser. Okay, so based on the last week's discussion, we thought we'll diverge towards more on dynamic hypergraph, because the graph was way too mixed up, because we had all the time points. So to segregate based on time points, we decided on making dynamic hypergraph. Now coming on that, so last time we saw 2 types of hypergraph, spatial and then lineage hypergraph. So in spatial also we created it in 2 ways. So one was by distance based metric, we did thresholding, and the other was clustering using dbScan. So that is what we did. So this is the first method by thresholding, and we have created a bunch of hypergraphs based on time points. So keeping a distance of 10, 10, 10, we have created these many hypergraphs. I think it's 4 into 5, 20, 19 hypergraphs. So creating 190 hypergraphs for every point, which is 190, was taking too much time. So for now, I created only individual hypergraphs after every time step. So it's also easier to visualize. And we can see that the number of cells are also increasing as we go time forward. But in the end, as we reach towards time point 190, it again gets quite crowded. So just a note here that in time point 81, we are not including those cells which were present at, let's say, time point 1 or time point 11. It only has cells which has time point which exist and which live at time point 181. So still it is quite overcrowded. Then then I created hypergraphs based on clustering, and as we saw yesterday, in the last week in the previous week, the hypergraphs created by clustering method were little less cluttered, unlike the previous method. So based on this method, I created again the hypergraph. So this hypergraph is created only for a single time point, which is t equal 1. And we see that here we have 4 unique cells. So one is EMS, then AB, ABP, and ABA. And we observe that for every cell, we have like multiple hyperedges, which looks a single cell is being incorporated by multiple cells. So trying to understand that, we did a little bit digging. So what we observed that at time point 1, we have total number of 24 cells, and those cells are like this. So cells are AB, AD, ABA, ABB. So we see that same 4 cells have like multiple instances of it. So AB is here, and so on. Similarly for ABA also exists at multiple instances. And when we observe these cell positions, so for example, at time equal 1 only, we are focusing on. So this AB cell has this set of XYZ coordinates and its size, and another cell AB, the same cell AB, but it has a different coordinates XYZ and also a different size, even though their mother will of course will remain same because it's originating from the same parent. But still looks like every this same type of cell also exists at different points. But when we visualize these cells in our hypergraph, we saw the overlapping of these. So this is because we are not actually drawing the coordinates XYZ. If like that, if we try to do that, then it will be a little harder to observe it because then we will have to do a 3D plot. But the goal is next that only. Anyways, let's focus here only. So that is why we saw that there was these many hyper edges overlap. So at this point, I made a decision like just to make it less cluttered, just to have a single hyper edge for every unique type of cell. What I thought was we can do we can just count the unique cells here. So at time point 1 we have these many cells, 24. So instead of this, we'll just count the unique count of it, which is like 4 here. And but we will also count the frequency of them. So if AB exists like 10 out of 24 times, I think that is not the case, but still assuming, then we can have a bigger set of bigger size of node for AB compared to like if EMS only exists twice, then we can have a smaller EMS. Let me know if that can be useful, just to see that AB is existing more number of times in that organism, in the C. elegans organism at that particular time point. Yeah, so I think that is what I did. So you can see at time and one more point I wanted to mention. So now since we have this AB existing multiple times, but we only want to take like single instance of AB, so these AB values which have like XYZ coordinates and size. So I averaged these coordinates across all different ABs at that particular time point, which is t equal 1 in our case for now. So now when will plot at time equal 1 AB? So this is how it will look like. So this so all these graphs. So at time point equal 1, this AB and all other cells also will have XYZ coordinates which are averaged for different instances of it at that time point. And we see now that we have a single hyper edge for every unique cell, right?
Yeah.
And that is what we did for time point 11, 21, and 31, and so on. And it's less cluttered and possibly will make more sense when we are analyzing. But another thing, as I mentioned, we can try is we can increase this node size based on how many instances of AB are present at that particular thing. Maybe it's useful, I thought, so just for visualizing purpose.
Yeah.
So I had a bunch of plans after this, in specifically dynamic hypergraphs. So what we can do is we can create like incidence matrix. So incidence matrix is how we mathematically represent the hyperedges. We have other ways also like incidence dictionaries. We can do that also. So it will help us view the number of cells, density, aspect ratio, etc. At different time points. So I think that will be quite useful. And then second thing is we can view the degree, neighbors, and other features, other spatial features for every nodes, or we can say hyper nodes in our case. So as we can see this AB, right? So this AB is a collection of a lot of ABs at that particular instance. So I assume loosely it can be called as hyper nodes also at this point. Please let me know about this also, whether I'm using it in the right way.
Okay.
And third thing is we can build sub hyperedges. So what right now we have is we still have like at higher time points we have still cluttered too many cells at every time points. So instead of like, let's say if you want to focus on a particular cell or particular connections which are originated either from spatial or lineage, we can build sub hyperedges or subgraphs. I have said it wrongly here. Sub hypergraphs. Hypergraphs. We can do this by restricting the nodes and the edges. And I was seeing the Hypernetics library, and we can do that. So that is how I got the idea from. And few other things then we can do is we can measure centrality measures for the graphs at every time point, and we can see the evolution of centrality measures. That will be interesting to see. I think. And then so this was something which I got idea from somewhere else, advanced tutorial of some library. So what it was saying is we can build the weighted hyperedges. So here we have hyperedges, right, across or for all the cells and maybe for across multiple cells at a time. So but we don't have any associated weights for it, not telling like how big or how strong that hyperedges. But we can in our case also we can create weighted hyperedges, and we can create that by the count of how many times this hyperedge has appeared at different instances. So for example, let's say a particular hyperedge has been existing for time point 11, 12, 13, and let's say till 21. So its weight can be of value of let's say 10 because it has appeared a lot many times compared to a single hyperedge which only existed for one or two time steps, and then it got like divided or segregated into further subpoints, or maybe the cells got like separated based on their movements. So we can have like a weighted hyperedges if it is required. We can model it that way. And then I was seeing the concept of dual hypergraphs. I should have posted the picture, but yeah. So I wanted to ask whether dual hypergraphs can be useful in our case, and I wrote it useful for lineage hypergraphs specifically. So to show what dual hypergraphs is, I will just open a tab. So dual hypergraphs. Yeah. So this should be a good visualization to understand what a dual hypergraph is. Is it a dual hypergraph? Yeah, this is a dual hypergraph. So what we are doing is, so initially we have this normal graph where we have V2, V3, V1, V2, V3 edges, and we have vertices, and we have then E1, E2, 2, and I think E6, E5 edges. So we just invert edges and vertices. So now we have edges as vertices and vertices as hyperedges specifically. So for instance, this V1 is connecting to three hyperedges, right? I think it's E1, E3, and E5. So now this vertex V1, which is now a hyperedge, will encapsulate three vertices, the three new vertices which we call as E1, E3, and E5. So these three. I don't remember why I thought it's useful for lineage hypergraph, but I'll get back to it. But if you get any idea like how we can model, how dual hypergraphs can be useful to model lineage hypergraph in a situation, then we can definitely try that.
Right.
And I have one doubt before we go next. So, should we increase size of the node based on the number? Yeah, I asked this before only. So, should we increase the size of the node based on the number of that kind of cell? Yeah, that point. Like if we have like multiple instances of this cell, should we increase the size of this maybe? Yeah, I have asked this before.
Okay.
So for now, for now that was it. I have a bunch of plans and I was exploring different libraries, so could only do this much.
Well, that's a pretty good update. Thank you, Mehul. It looks like a lot of good work. I like the hypergraphs by time point. So you generated all these graphs, and they're sort of not really dense at first, but then they become denser. And I assume we can explore each one if we find one that's, because it looks like, you start with like these cells on the edge, and then you start to get this thicker edge. And I'm just describing the sort of the topology with some cells in the middle of that sphere. And then so this is really informative. Then it's starting to densify in the middle and on the edges, and then finally it just becomes really dense. And I don't know what the structure is if we zoom in on each hypernode and look at how those, if we look at like kind of local structure. But I think this is really interesting because it does bring up that kind of, and we know from like in embryos that, we start out with maybe 2 or 3 cells, and then we start to get these sort of, meta patterns in the distribution of cells. So this is really interesting. So we might mine these, like look at these, look at the sort of the positions of the hypernodes, and there are all sorts of analyses you can do. And I'm not sure. I know that the standard network measures, and we could probably use those. I'm not sure what kinds of measures are state of the art for hypergraphs, but there might be some graph measures we can derive, and that would involve going to the literature and finding those. But that once you have the graphs, the description in terms of statistics, then we can actually, or in terms of the position and the connectivity, then we can do the statistical analysis on it. So that's good though that we have those. And then if you go down to your questions, can you scroll down?
Is it there?
No, it's frozen, I think.
Okay. Yeah. I'm already at the questions.
All right, there we go.
Probably it will take a few seconds.
Yeah, there we go. All right, so then the plan, we could use an incidence matrix, view degree neighbors. That's all like graph statistics. Then build subhypergraphs. Yeah, I think subhypergraphs is a good strategy. We can restrict the nodes and edges in different ways, and get like maybe clean up the. We might use like a more restrictive criterion, and see if it cleans up the topology a little bit. I mean, what I mean by that is just so that you can see things, the strongest relationships, say, and they're just working that method out to see what that looks like. And then let's see. So we have the dual hypergraphs, which I like this idea. I think it would be useful in lineage graphs. So in the lineage graphs, our nodes are cells. Our hypernodes are sort of cell categories or cell classes, whatever you want to call them. The edges are sort of the next generation, or if we have a round of cell division, those cells may differentiate, so they leave that category, or they the category doubles, so the number of cells doubles, so that cell becomes 2 cells, so there's a connection from 1 to 2 and all that. So those are our edges and our hypernodes. And then if we invert those, if I'm understanding this correctly, that might be interesting. We might be able to actually have I guess, nodes that would describe those division events or those differentiation events. And then, the edges would be I guess, the cell types or categories. I'm probably getting it wrong, but the point is that I think it'd be useful to describe some of those relationships in different ways. So, we know kind of the structure from the lineage tree. It's pretty straightforward what that is. But if we invert those relationships, we can actually find out, some, especially since you're sampling the graph at time points that aren't naturally division events, which, is probably good because we're not really interested in maybe the division events per se. We're interested in the relationship of or the state of the embryo at that time window. So whatever's there at that time window, that's what we want to know. And the doubling events will be captured in that. So that's good. Yeah, so I think there's some application. I'll have to think about it a little bit. The last one, the question: should we increase the size of the node based on the number of that kind of cell in the same time? So yeah, that might be useful to have like a map where you show like the relative contribution. So like, the data that's collected on these are, they'll sample the cell at different times. And theoretically, the longer the cell's around, the more samples you'll have. So like if a cell lives for 20 seconds or 20 minutes, or let's say it lives 20 minutes versus 10 minutes, there should be more instances of that cell in the data set. I'm not really sure how that works out in their sampling scheme, but basically, I think it should be overrepresented. So, it'd be nice to know kind of that information about what cells are over, because we tend to average out over all the cell observations. And that's good for kind of a, mean description, but there's a lot of information in there maybe we're not picking up. So like if I take the mean of something that's been everywhere in the embryo, the mean position will be in the middle of the embryo. That's not very informative. So we need to have something that's a little bit less smoothed over. We need to have some information about the variation that you might see as well. But not noise. Right. Yeah. Okay.
Okay.
So yeah, that's very good. That's very good, and I think that I think you're going in the right direction here. I think that there are things you can play with, but I think once you have the basic scheme down, then we can do these things like the graph statistics and playing around with the layout and with a lot of network science. The layout is kind of a thing unto itself. So there are a lot of different types of layouts that people play with. There's like the hairball, which is what you're showing, and then there's like force directed graphs, there are ordered graphs, all these other things that we can do. But we have to get the basic relationships right first, which you're seemingly getting done. So.
Okay. Yes. Thank you very much.
Thank you. Yeah.
Okay. So for this week's plan, I am thinking of as we discuss this plan, I'll try to do 1, 2, 3, 4, and probably 5 as well. I'll explore dual graphs a little later. But most important thing which I'm thinking of right now is exploring lineage hypergraph, and which will then lead to dual hypergraphs also if we think it's useful. Yeah. So I hope this sounds good for the week, for the upcoming week.
Okay.
And right now I feel like we are diverging too much. That's not bad. We are exploring the nature of hypergraphs and which hypergraph modeling will be will be useful for our data case. But after couple of weeks, I would like to go into specific 2-3 kinds of hypergraphs so we can model both the spatial and lineage hypergraph as good as we can. So yeah. So we can conclude well in the end.
So from your explorations, do you think the clustering or DB scan method is better?
Yeah. So okay, right. I have written it down now. in the previous notebook, which we saw. This was the DBScan method. Right? So a few methods, a few features I have written about DBScan is. So one feature is that we don't have to specify the number of clusters. So what happens in k-means is we have to specify beforehand without knowing even the topology of how cells are oriented spatially. We have to tell that we want this many number of clusters, and this actually adds up to one of hyperparameters which we have to decide, which we have to come up with. So DBScan avoids the need of this. So the first point is that, and the second is it is quite robust to outliers. So what we can observe is like at time point 1 we saw all the 4 points were quite divergent at a different places. So so possibly all could look like outliers in the beginning at beginning time points, a lot of cells. So DBScan is quite robust to outliers. So what it does is it basically does not assign those points to any cluster. So we saw that going to the same notebook before. Give me a sec. Yeah, I'll wait for like few seconds. So the screen is static.
Okay.
Yeah. In the I hope it should be there now.
Yeah.
So at time point, yeah. So at time point 1, we had these cells, right? A B A B A B A. So we saw now we see the cluster indices of it. So I did not explain this part. So we see that it is -1, -1, -1 for all the cells. So what it means is that these cells are not being assigned any particular hyper edge or cluster, because we are doing clustering here, because all the cells are quite far away right now from each other spatially. So DBScan allows us to do that. It allows us to deal with outliers in the way that it won't assign them to any clusters. On the other hand, k-means will force, or even other methods will force the cells, every cell, even though it's very farther from from the closest hyperedge, to force them to be a part of it. So here DBScan is useful. And then coming to the third point, it is capability to find arbitrary shaped up clusters. Yeah, this is very important. So this is specifically the k-means drawback. So k-means only can assign spherical structures when it's creating the cluster. It doesn't deal with, like, for example, if we say ring-like cluster and in between there is some different cluster. So DBScan, but DBScan does allow that. I can show a picture, but is it clear, like the third point?
Yeah.
Okay. Yeah, so that were the three reasons DBScan could be more useful specifically on our case because of how our data set is. And we have a few hyperparameters associated with it, which is epsilon and the min samples. So epsilon says, so how many, epsilon is basically the maximum distance between two samples for them to be considered as part of the same neighborhood or same cluster. So this I have assigned it as 5 only, like we have done in the previous threshold 5 value. If we do 50, then it is too much. And min samples, so like to consider a hyperedge, to consider a cluster as a hyperedge, or to so this is the minimum number of samples. So I have put it as simply 1. So a single node or single hypernode can also be treated as a cluster on its own.
Yeah. And then you have min samples. Yeah, it's basically like, almost seems like k-means is forcing us to explore the spherical cow. I don't know if you know that expression, but and then DBScan allows us to sort of have more sort of shapely clusters, categories, but it doesn't assign a cell to any one category if it doesn't think that there's a category to assign it to. So that's both good and bad. I mean, I guess we could just treat those cells as individual points, and then start to get hyper nodes. But so yeah, this looks good. This is so this looks like DBScan might be good. I would just say probably it's a trade-off though. I mean, we could kind of go with DB-scan, but then also, kind of have k-means in the background. If there are things we can't get to with DB-scan, we might try k-means as an alternate way of finding clusters. And, because you don't always want to try the same method for everything. Because this is a kind of a tricky data set. You have like the spatial heterogeneity, this temporal heterogeneity, this temporal growth, and then you're trying to sort of describe all these different, I guess, sublineages of cell with the same model. So it's really hard to do with one method, yeah. But this looks very good. Thank you for this update. I think this is very informative. So yeah, yeah. So I mean, the next week you'll probably kind of work on maybe some of the graph statistics, or kind of polishing up some of your exploration or stuff like that.
Yes. Yeah. But before that, you mentioned a point like I have doubt on. So what you mentioned was like we have this multiple instances of AB when we were constructing, right? At time point 1, we have number of cells 24, and we have multiple instances of AB. And what I did was I I averaged the coordinates X, Y, Z. So you said that even if we have like cells AB at 4 different corners, but if we like do averaging, then it won't make sense because it will fall in the center. So right now I was following that, and what we got is a graph like this. But let's say if I don't follow that, I mean, it's wrong to follow that, as you correctly mentioned. So how should we create spatial graph in that way? Because if we don't average them, then we will have to concatenate them like in a fashion that we have 4 set of different nodes of the same type. But we also don't want to make like this set of a graph like this where we have like multiple hyperedges across across a single cell. I mean, it is okay, but it's just for visualization. I try to get a different method.
Yeah. So I mean,
should we proceed with?
Oh, so yeah, there are different ways you can do an average too. So you could do a weighted average, which means that each one has some contribution to the overall mean. Or you could have like, I don't know. It's an interesting question of how to do that. I'm not really sure. I mean, you could have like a centroid, which would be like this mean, which is basically a centroid of all those cells. And then you could weight them by sort of their variance. So like if your average turns out that you have a lot of variation, like if you have cells at like 4 points on the edge of a sphere, and then you end up getting like a value in the center of that sphere, and that sphere is very large, you might weight because they're not going to be evenly distributed. You can weight them accordingly. Or you can, maybe do some sort of thing where you create a hypernode of all the cells of that type. Like all the AB cells are in a hypernode. The hypernode has like a centroid. It has variation. And, you might actually just treat the cells themselves as hypernodes with all their instances inside. And then they have, edges with other hypernodes, which are other cell types. And you could do that too. I mean, there are a lot of ways you can treat the individual cells. I would just, the base model would be the averaging, the simple averaging. But if you can think of a way to do like a weighted average, or some sort of centroid model, or even like where you just have a hypergraph of all the instances, that might be useful too. Because I'm not really sure how the sampling, when we collect the data, we sample the embryo based on just these kind of observations. So there's no evidence that one observation is better than any other, but it's also the fact that they're all independent observations, so they all may have some value in telling us something about the structure of the embryo. So I know that's kind of a hard answer to your question, but it's that it's just that's the way that's what we have in terms of our data set.
Yeah, yeah. But that is an interesting feature of dataset, I think.
Yeah,
So
yeah.
I think so Hypernode, I think it's a good idea. So somehow we can put all the information by telling the variance that we have a lot of variance and different in their positions, but still we have this Hypernode with this averaged sort of like weighted averaged hyperedge. Hypernode, I'm sorry.
Yeah. Yep. Yeah, that's good. Well, thank you for that update.
Yeah, thanks.
All right. So I actually mentioned the spherical cow before, and I don't know, people are familiar with that metaphor, but so there's an old joke about a physicist who is commissioned to study like milk production and cows, and then they say, okay, how do you start solving this problem? And then they say, assume a spherical cow. And of course, the joke is that in physics you do things as spherical models. You take things and you abstract them away to the point where they're not really like they don't have any practical value, but you can actually find theoretical value in those models. And I think there's a variation of that joke with an economist, because economics does the same thing, where they have they abstract the real system so much that they have this model that's mathematically tractable, but not doesn't have a lot of connection with the real world. And so that's always a joke that people use to sort of make the point that our theoretical models are a lot of times divorced from the real world models. But the other point, the counterpoint, is that there's this theoretical value in these kind of models that you don't get with just a simple data analysis of the real world data. And this is why, because our real world data is oftentimes so messy and there's so many variables, variation present, that we can't really do anything useful with just this straightforward analysis. So now I'd like to go over something I mentioned in the meeting, and that is to talk about the average position or the centroid of a cell. It's been observed multiple times. So let's take AB as an example. So we have AB, and we can observe this over a number of time points. We might observe this from T1 to T2 all the way to, say, 20 time points, T20. We can get independent observations at each time point. AB is either present or absent in the time point that we're looking at. So usually when we get data, we get microscopy data with sampled at specific time points, and then that image either has that cell or does not have that cell. So our image, of course, is going to capture that cell at any one time. And of course, the cell can move around. It can be in different locations in the embryo. It's maybe an imperfect method for getting this information. In any case, this cell persists until we get a division event, after which ABA and ABP exist, and they exist for a time. We have the same problem with ABA and ABP. So we have this temporal variation. But what's more important are, when we have, we're looking at crossed organisms, or crossed embryos, and in those cases, we can have instances of observation. So if we see AB in one embryo and AB in another embryo and an AB in another embryo, they'll all have their own three-dimensional position, and those are independent observations. And those observations will persist until, again, there's a division event, and then we can look at ABA and ABP. In general, we think of these as independent observations. So this is the symbol for an independent observation. And AB nominally is independent of AB. So any one instance of AB is nominally independent of any other instance of AB. We could actually, track the cell over time, but because it's hard to really capture that time evolution, we consider it to be independent. Now, we could have a situation where in the time series where they're dependent. So AB1 is autocorrelated with AB2, so is the position changes in time, that represents true migration. On the other hand, it's hard to do because we have different frames of observation. So this is something that we have to keep in mind. And then we can also have a statistical summary of an average or a mean. So the mean of AB is the mean of all these instances. And so we can have these independent observations, we can have these temporally dependent observations, or we can have these mean observations. And so when we have a mean observation, we have something like this, where we have a point cloud for AB, and that point cloud consists of a bunch of cells. And those cells can sit in different positions. Sometimes there's quite a lot of variation around them, and they sit in this point cloud. We could define this point cloud roughly like this, and then we end up with a centroid position. So this is a centroid for AB, and the mean description is the centroid position. But as you notice, we're filtering out a lot of variation. Now when we move to the division case, AB, or that centroid, will divide into ABA and ABP. ABA and ABP again can be represented as these point clouds, and again with a lot of variation around the centroid. So our model of sort of cell division is that we take the centroid for AB, we take the centroid for ABA, and the centroid for ABP, and it's a nice, neat sort of set of lines that diverge. But in reality, you have a lot of overlapping volumes with overlapping cell positions across all these independent instances, or sometimes temporal instances. And so this makes it a little harder for us to understand what's going on. The centroid model makes it easier to sort of represent it, but there's a lot of variation underlying that isn't described. So this brings us to the thing I was talking about in the meeting, having a weighted average. So your weighted average is basically where you have this AB point cloud, you have these independent observations of cells, and you have the centroid. The centroid value is the mean of these cells, but we don't really want to just take the mean and leave it at that. We might want to take a weighted average. And the weighted average would be where we take each cell and has a certain weight relative to, say, a centroid. So like in the k-means example of what Mehul was talking about, you might define a centroid position. And then you might say, okay, how far away is each cell from this centroid position? So you derive a centroid position a priori. You then fit the cells to the model of the centroid, and then you derive a new centroid prime, which is, the value of this. So it would be like AB mean prime, which would then be the new centroid, the weighted average. And that gives you a little bit more refined information about this point cloud. So this is a point cloud of points for an individual cell, and in the embryo we have this point cloud of points for all cells. And so thinking about the hypergraph case, we have these hypernodes which contain nodes. These nodes are individual cell IDs. They can be averaged. Now we can also do a hypernode for individual. So this is for the embryo or for cell types. And we can also do this for a single cell type. So this is the hypernode for AB. Then you have a bunch of ABs within this hypernode. And then they connect to other hypernodes. So you might have, say, for example, ABA and ABP. And that represents all the hypernodes here. So we can actually look at how these are connected. Like this. We get information about how all these individual cells kind of divide. So we'll have this mapping from one hypernode to another. It's basically this trivariate graph where you have the hypernode for AB, mapping to its daughter hypernodes. And you have these correspondences that tell you something about the division events, and they're all going to be different. So we would take the information for each instance of observation and build a division event, build a branch of a tree, and then we can do what we want with that. We could average those. We could describe those in terms of modes. So sometimes, you might have clusters within a hypernode or clusters within hypernodes. So, these cells over on this part of the hypernode ABA map to these parts of the cells in the hypernode AB. And so we can say that there's like some spatial, in some of the observations there's some spatial bias. So it gives us a better account of this variation. I just wanted to mention that thank you, Mehul and Paki, for your GitHub contributions.
Yeah, thank you. I saw that it's merged now.
Yeah, so this is the DevoGraph repository, and so we've been active with pull requests here. So I have accepted several pull requests this past week. Mehul pushed his data exploration notebooks, and this is number 21. Number 20 was KMAP, the KMapper implementation by Paki. That was all the stuff that she presented last week. And so those are two that have been pushed. Thank you for your contributions, and hopefully, we get more as we get on with GSOC. And I would encourage people to push, if you have maybe every week or every other week, just to have the habit of pushing things to the repositories. So those contributions are live in DevoGraph. They're part of stage 2. So if we go to stage 2, we have the TDA using KMapper, and then we also have the Hyper DevoGraph directory, which has a lot of those things in it. So that's where those things live. Okay, that's good. So I also wanted to mention that the GECCO conference is this week, where it's starting up imminently. And GECCO is a conference on genetic algorithms. So if you're interested in genetic algorithms, there are a couple of conferences. There's EvoStar, there's A-Life, and then there is GECCO. And GECCO is more sort of the applied genetic algorithms conference, where people do things just kind of like tweaking the algorithms or, applying them to different problems. Whereas A-Life is more maybe the theoretical side of that. And then EvoStars is kind of like a bunch of stuff that people are doing. It's, so GECCO this is GECCO. It's being held in Melbourne, but it's a hybrid conference this year. The full title is Genetic and Evolutionary Computation Conference, and their logo, not surprisingly, is a gecko with a double helix overlaying the gecko, and that's not to scale, of course. So, but that's why they call it GECCO. And so, yeah, they're holding in Melbourne. There's, it's a pretty big conference. You have a lot of talks. It's one of these conferences where it's pretty overwhelming because it's of reasonable size. So you have to plan your route through the conference. This thing here is this tutorials page. So one of the interesting things about a lot of conferences is they have these tutorials, and people will host tutorials on a number of topics. Usually these are very useful because they give you some depth in a certain area that you wouldn't be able to, say, take a class on, or it might be something that you see in papers again and again, and you don't really understand the method as it's described in the paper. So they'll have these tutorials where they walk you through a method, they give you the software, they run a set of problems, and then you get some insight into your into some method in the field. So they have a lot of things here. They have robust optimization. So genetic algorithms, of course, one of the foci of genetic algorithms is optimization and creating optimization schemes for data. And, this is, of course, a longstanding interest in the field. So there's robust optimization. There's this gentle introduction to theory for non-theoreticians. This is, of course, just talking, let's see, this is about sort of theory. This doesn't discuss mathematical methods or particular results, but it talks about what the theory of evolutionary algorithms aims at. So there's a theory of evolutionary algorithms, not just the implementation. There's theoretical research in evolutionary computation, and that's separate from sort of benchmarking and those sorts of things. Then they just kind of tell you how to deal with theory and how to formulate theory and what are the trends in the community. So this is looks like an interesting workshop or tutorial. They have Bayesian optimization, and this is, of course, in the context of genetic algorithms. So one of the strengths of evolutionary algorithms is that they can be applied to black box optimization problems. And so this is where, we can use these methods for a lot of things other than just kind of modeling evolution or, looking at that problem in and of itself. So Bayesian optimization is actually something that we can model with genetic algorithms. It's very amenable to it, and that's what they're talking about here. Bayesian optimization has a lot of applications to deep learning and design optimization, or even stochastic optimization, something we use in operational research. And so this is where you have to build features into your genetic algorithm, some sort of Gaussian process, and have a memory for previous evaluations, and you put those together into a Bayesian model. So this is an interesting thing because, we often talk about Bayesian methods, but sometimes they kind of are separated away from, they're abstracted away from the problems that they're trying to solve. I don't have time to give concrete examples, but just suffice it to say that people throw the around Bayesian around a lot, and the insights are not as obvious as the sort of the promise of the method. So this is nice that people are kind of working out the details of how to implement this in code. There's coevolutionary computation for adversarial deep learning. So there are some applications of deep learning. There's the evolution of neural networks, which is interesting from the standpoint of what we're talking about with neural developmental programs. So this is another kind of approach to evolving artificial neural networks. So, you can take there's this paradigm called neuroevolution, where people build neural networks and they evolve them to sort of be specialized for certain tasks. It's different from training a model in a number of interesting ways. And so, as they point out here, deep learning performance depends crucially on the network design, i.e. its architecture, hyperparameters, and other elements. So while many such designs are too complex to be optimized by hand, neuroevolution can be used to do so automatically. And so this is what they call evolutionary auto machine learning. And this is where you can get good deep learning performance, even with limited resources or state-of-the-art performance. So this is one of the goals, of course, of trying to build deep learning networks, is to build them in a way that doesn't require huge computational resources. And so, there's a lot of focus on stochastic problems and stochastic solutions and things like that. Evolutionary computation is amenable to that because, of course, evolution works on the principle of stochastic generation of variation and selection. So we can actually select from a stochastic generator, and we can get, we know that theoretically this should work. So it's very amenable to those types of problem spaces. And again, optimization is a big deal. There's also evolutionary machine learning for interpretable and explainable AI, which is a hot topic now, but linking evolutionary algorithms into that deep learning space, machine learning space. Evolutionary reinforcement learning, which is an interesting area because people have been, kind of getting into reinforcement learning after kind of a lull in the field. And so there are a lot of ways that people, kind of interesting ways people have approached reinforcement learning. And so in this case, they're looking at evolutionary approaches. So, reinforcement learning, we've been able to do things with, training models in video games like Atari, StarCraft, chess, and Go, which are board games, but we still can do that. We can train reinforcement learning models to play those games quite well. And then, we can actually look at other types of problems like robotic control, high-speed drone control, and controlling something like a plasma reactor. So very complex systems, collective behaviors that exhibit collective behaviors or complex dynamics. And, people are even applying reinforcement learning to things like chatbots. And so that's interesting. And then, so interestingly, elements of evolutionary computation, such as population-based training, which is a technique from Jaderberg 2017, and quality diversity algorithms from Pew 2016, represent key elements in some of these breakthroughs. The combination of evolutionary computation and reinforcement learning methods is not new and has gained more popularity and interest recently as researchers discover the limitations of the individual approaches. So this is, there are a bunch of outstanding questions here. There's curriculum learning and environment generation. There's neuroevolution, meta-learning with evolution, quality diversity, which is a sort of technique that I'm not going to get into right now, but it's a popular technique in evolutionary computation. And then other combinations of evolutionary computation and reinforcement learning. And so, then there's some things very specific to genetic programming, model-based evolutionary algorithms, robot evolution, which is interesting, representations for evolutionary algorithms, which is important in terms of how you build the representations themselves. So evolutionary algorithms really take a problem and they model it as sort of something that's on an artificial chromosome where the problem is encoded in the genes of the artificial chromosome. And that genotype then describes the problem. And you basically can evolve the problem space by mutation, recombination, and selection. So you have these different things that are operating on this problem space. And the idea is that you can maybe recombine the problem space or mutate the problem space in certain ways, or at least the representation of problems in that problem space, so that you can reach optimum. And so this is kind of what they're getting at here, where you build these representations more or less effectively. If they're more effective, it helps you with your solutions. If it's less effective, then it's harder to run the algorithm, get an insightful outcome. So successful and efficient use of evolutionary algorithms depends on the choice of genotype, the problem representation or the mapping from the genotype to phenotype, which is basically the representation in binary values that make up some sort of gene or make up some sort of chromosome, and then modeling that as an outcome in the problem space. And on the choice of search operators are applied to genotypes. And so they just kind of get into this architecture and how to design things and how to implement them. Then there are some other interesting, there's of course the obligatory large language models application as well. So that's the last tutorial. A lot of interesting tutorials. This workshop actually is really interesting. This is the graph-based genetic programming workshop. So this is about it's not graph neural networks, it's graph-based genetic programming. So it's GraphGP. And we talked about, of course, graph neural networks. We're talking about hyper networks and graph neural networks. This is something that really kind of gets at this idea of representing your problem and having this good representation, but taking it to another level and applying it to graphs. So the evolution of graphs, basically. And so, they talk about this. Well, the classical way to represent programs in genetic programming is by using an expression tree, different genetic programming variants with graph-based representations have been proposed. Graph-based representations have led to novel applications of GP in circuit design, cryptography, image analysis, and more. This workshop aims to encourage this form of genetic programming by considering graph-based methods from a unified perspective and to bringing together researchers in this subfield of research. And so this is just kind of a call for papers, but they do have a program now which kind of talks about some of the things that people are going to be presenting on. So there's this idea of using evolutionary model merge. This is a general method that uses evolutionary techniques to efficiently discover the best ways to combine different models with diverse capabilities, using ideas similar to graph-based neural architecture research. So this is sort of the evolutionary version of this. And this is based on this work by Sakana AI, which of course is a group run by David Ha, I think, or at least he's involved in it. And this is where they're building these evolutionary models that are relevant to machine learning. So Sakana AI is being represented here. Evolutionary Robustness by W.B. Langdon. He actually presented at the EI workshop, the Embodied Intelligence workshop this last year. And I think this is similar to the work he presented there. This is on, of course, genetic programming and other types of evolutionary algorithms. So there are different types of evolutionary algorithms. Genetic programming is a subset of that. And, talks about how those methods have demonstrated creativity, which is something we talk about with respect to deep learning and machine learning, but is something that's very closely tied to genetic programming and that technique of, modeling a system and being able to mutate it and recombine it in different ways. And so this kind of talks about kind of how, there are these results, the GP is quite popular. And then this talks also links to these long-term evolution experiments of Richlinsky and sort of how evolution can generate a lot of creativity, a lot of variation in running for a long time. So this is kind of talking about how, in genetic programming, populations tend to converge, but in real evolution, populations don't tend to converge. And so there's this gap between sort of our real-world systems of evolution and the in silico aspects of evolution. So it talks about mutation testing, which is a technique for kind of looking at software being robust to different changes. So if you mutate software, can you continue to run that software, or does it crash and burn? And so this is something that genetic programming or evolutionary algorithms can provide insight into. So this is an interesting area of research. And then, of course, this mangrove architecture, which is where, if you're looking at sustained innovation or innovation over time or keeping innovation going in a system, you can use these mangrove architectures, which are composed of many small trees, which are intimate with their environment. So this means that you have these local trees that sort of characterize local variation in the environment, and then they're put together like a mangrove tree, which is kind of like joined together using a common root system. So this is an interesting kind of innovation here. This is citing some work by the author. And yeah, we won't get into that. Looks like that's perfect. Okay, so yeah, then there are some other oral presentations on different methods, directed acyclic program graphs, search trajectory networks, and some other things as well. So a lot of interesting methods there and techniques. And of course, evolutionary algorithms are one approach to dealing with that complexity that we see in our data sets. And, it'd be interesting to see how that relates to some of the work we've been doing with graph neural networks and some of these other things with developmental trees and other things.
I think some of the techniques are being applied to the tensegrities.
Okay.
Like I'm recognizing some of the similar language.
Yeah.
Genetic algorithms.
Yeah.
Yeah, so they're applying it to tensegrities.
Okay.
Yeah, I'm not sure how successfully, but and I'm not into that field well enough. Like I don't.
Yeah.
In
No.
other words, if I attended the conference, I probably wouldn't know what was going on.
Yeah, it's very specialized. But yeah, and the techniques they can be slow to kind of pick up. Like with a machine learning algorithm, you can train the model, and sometimes you just have to overtrain it and really have a lot of data specified. But with genetic algorithms, you really don't necessarily need to train it, but you need to represent the problem well, and then you need to be able to run it for a long time to get a solution. And so like I could take like a tensegrity structure and map that problem space to like an artificial chromosome. I could mutate it to see, if I change things, does it still work? Is it? And then there's a selection function.
It's called form finding.
Form
Find
finding.
the
Okay.
best optimal form.
Yeah. That sounds like something that they've done a lot of things with like.
It makes the whole thing more involved and difficult. Yeah, form finding tensegrity structures based on force density method. Well, oh well. Anyway, definitely there was a genetic algorithm kind of technique. I don't know.
Yeah. Well, I mean, I know they've done a lot of stuff with robotics and looking at the shape of robotics and sort of modeling, how do you evolve different morphologies that are optimal. So, you want to have a morphology that, can walk, or, a morphology that reaches for things, or a morphology that does, like rolls over rough terrain. So you can evolve different solutions for that from your representation. You can mutate it, recombine it, and then say, I'll put a selection, I'll put a selection operator on it and say, find the best phenotypes for the score, and then it generates a bunch of variation into different solutions, and then you can test those. You could model those in the real world and test them. And so but the thing is, it takes a lot of effort, in terms of computation, in terms of getting the representation right, and then doing the testing in the real world to see if it actually gives you what you want. But the idea being that you generate these options that are, pretty good, and then that's kind of what you want. And it has this creative aspect that machine learning doesn't have, and that it gives you this option. It doesn't focus on kind of like optimizing the model straight away. It focuses on giving sort of the best solution. So you can do optimization, but that's not really the goal overall. Overall is to evolve different variants that are optimized, and then, and people approach this in different ways. So some people are just interested in sort of engineering optimization, and that's it. Other people are interested in the creative aspect and generating, multiple solutions for something and seeing what those look like. So.
Oh well, can you hear me?
Yeah.
Okay, that's nice. My screen froze.
Oh. Okay.
And my computer will freeze after a bit. I seem to have a problem.
Yeah. Okay. So I don't know if Morgan had anything to say or.
No, just listening in.
Okay. Yeah. All right. Well, thank you for attending, and thank you, Mehul, for your update, and see you next week.
Bye.
Bye. Okay, now I'd like to talk about a few papers. These are papers on C. elegans development that are really kind of relevant to some of the things we've been talking about with respect to noise, position of cells, and sort of, and then looking at lineage trees and some of the metrics we can use for those. So they're all connected to this discussion today about hypergraphs. This first paper is on novel metrics for lineage trees. So we've done some work on this where we've looked at mapping lineage trees to differentiation trees and quantifying a lot of this. What they're interested in here is they're interested in looking at new novel metrics for C. elegans lineage trees, which are of course easy to study. And so, they introduce some methods here that are really intriguing. So the abstract reads, high throughput experimental approaches are increasingly allowing for the quantitative description of cellular and organismal phenotypes. Distilling these large volumes of complex data into meaningful measures that can drive biological insight remains a central challenge. So one can resolve phenotypic measures for single cells onto their lineage history. So what they're doing here is they're interested in taking single cell genomics and those types of data and mapping them onto the lineage history of cells. So we're actually looking at phenotypic measures for single cells, but we can also look at single cell measurements of next-gen sequencing. So one can resolve phenotypic measures for single cells onto their lineage history, enabling joint consideration of heritable signals and cell fate decisions. So another thing you can do is take heritable signals. So if something exists in a mother cell, it can be inherited by the daughter cells. And that may have some relevance to cell fate decisions. So a mother cell can transmit things to their daughter cells. Those daughter cells then can differentiate. And we want to know what those signals are. So this is an interesting point about how we might do that within the context of a lineage tree, because it can really target some of those signals to specific cell fate decisions. Most attempts to analyze this type of data, however, discard much of the information content contained within lineage trees. In this work, we introduce a generalized metric, which we term branch edit distance, that allows us to compare any two embryos based on phenotypic measurements of individual cells. This approach aligns with those phenotypic measurements to the underlying lineage tree, providing a flexible and intuitive framework for quantitative comparisons between, for instance, wild type and mutant developmental programs. So when you have a wild type, which is the default genotype in a certain in an individual or in a standard lineage tree. And in C. elegans, you see this as a standard lineage tree because we do have a standard lineage tree that we have for wild type genotypes. You have the type of genotype that we have characterized in our data sets and so forth. Mutant developmental programs, however, produce lineage trees that are incomplete. Sometimes entire sets of cells are missing. Sometimes the timing is different of division. So we have mutant developmental programs, and we can actually look at how those mutant programs, or at least say gene expression profiles for those mutant programs, or other types of measures, can map to discrete changes in growth, in division rate, and in the presence or absence of certain cells. So in C. elegans, we can look at this quite directly using the lineage tree. We apply this novel metric to data on cell cycle timing for over 1,300 wild-type and RNAi-treated C. elegans embryos. So RNAi is RNA interference, so they use that to knock down certain transcription factors. You'll put in an antibody and it'll knock down the transcription factor. And you can, or you have a reverse transcriptase RNA that can knock down the transcription factor and turn it off, essentially. And so we can do this this is sort of generating a mutant phenotype. Our new metric revealed surprising heterogeneity within this dataset, including subtle batch effects in wild-type embryos and dramatic variability in RNAi-induced developmental phenotypes. So this is where we can look at some of this variability and how these RNAi treatments can really kind of change the nature of a lineage tree, all of which have been missed in previous analyses. Further investigation of these results suggests a novel quantitative link between pathways that govern cell fate decisions and pathways that pattern cell cycle timing in the early embryo. Our work demonstrates that the branch edit distance we propose and similar metrics like it have potential to revolutionize our quantitative understanding of the organismal phenotype. So this talks about lineage tracing in C. elegans, and of course, this is one of the main organismal models in which, we really can learn a lot about lineage trees because there's a tight mapping between lineage tree and this deterministic cell fate and the timing of it. So it's usually deterministic across individuals, and this is especially important when comparing like a wild type to a mutant. So we can look at the effects of mutations quite directly, and it gives us a very strong signal. So they define a metric on spaces of cell lineages. So they have to do this by kind of taking the lineage tree, thinking about the organization of that lineage tree, which is anterior to posterior. So in other words, the lineage tree unfolds where the left-hand side is anterior and the right-hand side is posterior. And so that gives us an orientation, sort of an anatomical orientation to this lineage tree. And so we can use this to sort of derive these sort of tree edit distances. And so this reminds me of some of the stuff we were doing with differentiation trees, although we didn't do it in the same way. They're actually doing this graph edit distance from graph theory. And so they derive this measure called tree edit distance, which is based on counting the minimum number of operations, such as adding and or removing a node or edge that is needed to convert one tree into another. So in differentiation trees, we are reordering the tree and figuring out some of the local aspects of that reordering. In this case, what they're doing is they're reordering the tree by adding or removing nodes or edges that can allow us to convert one tree into another. So you're just counting the number of changes you need to make to change the tree to something else. And that could be a mutant phenotype, or it could be an incomplete phenotype, or whatever. And so, this is a good way to do this. This sort of reminds me also of phylogenetics, where you kind of do these kind of operations on gene trees or species trees. And so I think there are a whole host of computational techniques you can do with lineage trees that are really interesting. This tree edit distance is a very powerful tool. It essentially reduces the calculation in determining the number of nodes that are different between two trees, a notion similar to other measures. So this is their figure 1, and this talks about these distance metrics, or these versions of distance metrics that they kind of give in this paper. And so we move from this tree edit distance to some other possible distances that we can calculate on a lineage tree. So the first one is tree edit distance, of course, which we talked about. This quantifies topological differences in nodes between two trees. And so we have tree NX and tree NY. Tree NX, of course, contains these cells. Tree NY contains a different complement of cells. And so when we knock certain cells out, we can get a different distance. We basically look for the change between NX and NY, and we reorder it, and we knock out cells, and we get this sort of distance between the two trees. So that could be APA and APP, or AAA and AAP, and those are the missing cells in each tree. So NX has some missing cells, NY have some missing cells, or NY might have cells that aren't in NX, and vice versa. And that's their distance. So they're looking at distances in terms of cells. We did some experiments a long time ago where we looked at the information content of lineage trees. And this isn't the same thing as edit distance, but it's interesting because, again, you're using you're reordering the tree. We were reordering the lineage tree into a differentiation tree, and then calculating the information content of that transformation. So this is a very similar thing, but it's interesting how you can play with lineage trees and get these kinds of information out of them. The second is intersection branch distance, or IBD. And this is the L squared norm between shared nodes value of trees. So this is where you look at the union between NA and NB, and alpha and N beta. And so you're just basically intersecting two trees together. You're looking at the L squared norm between these shared node values. So you have these shared node values between these two trees, alpha and beta. And then that gives you Vx and Vy, or these values are Vx and Vy. And then this distance, this intersection distance, which is the norm of x minus y. And so that's the way you get that. And so this kind of shows, I think, what this looks like. So you have these numbers on the branches and these alternative orderings, and you look at the shared branch or the shared node values. The third is union branch distance. So this is the intersection. I'm sorry. This is the intersection. This is the union. So this is looking at the intersection between alpha and beta. And this is the union between alpha and beta. So this is where you have N alpha and N beta, the union of those. And this is where you have the L squared norm between all node values of trees. So this is actually instead of just taking the shared nodes between these two trees, you're taking all node values of trees and you're uniting them. And I think the point here is to say that the intersection and union are complementary. So the union is everything, the intersection is just those things that are shared between trees. And so that's actually kind of like an information measure, but it's not quite an information measure. It's just kind of a different way of looking at it. In any case, what you're doing is you're comparing tree X to tree Y, and you're getting information from each one, and then you're kind of, making a statement about what's shared, what's not shared, and so forth. And so their figure 4 summarizes the union branch distance and how it reveals heterogeneity in the RNAi cell cycle timing coordination. So basically the difference in the wild type versus a mutant, which is represented by these RNAi experiments, is that it changes the cell division time. And so this is something that we can look at in terms of cell cycle timing. And so this actually takes the union branch distances and creates this heat map that shows the difference between the mutant and wild type phenotypes. So this shows you in minutes from 0 to 1400 these differences across these different categories. And so this is the summary here. You have the wild type embryos. You have this tree edit distance to wild type. You have these RNAi embryos. You have the intersection branch distance to wild type. And so that's what this is. And then you have this comparison of tree edit distance and intersection branch distance for RNAi embryos. So looking at those mutant embryos, you have a tree edit distance here, which you're getting in this B, I, and III. And then on the y-axis, you have mean intersection branch distance, which is what you have in II and IV. So in 1 and 3, you have this x-axis, and in 2 and 4, you have this y-axis. And so this is a comparison here. You can see that there's a cluster down here with a smaller cluster over on this side. So for tree edit distances near 0, there's a cluster. And for tree edit distances between 100 and 200, there's another cluster, with some outliers going in this direction of a larger tree edit distance. But you can see that there are two groups here clearly. The mean intersection branch distance doesn't really predict very much. Both of these clusters are at about a very low mean intersection branch distance. So that means that the tree edit distance is predicting most of this variation. Okay, so the second paper, and this is quite a lengthy paper as well, this is deterministic nature of cellular position noise during C. elegans embryogenesis. And early in the meeting, I kind of went over some drawings of what I meant by cellular position variation and maybe some of the strategies to control this in our data sets, just of cell position. What this is, though, is it talks a little bit about the deterministic nature of this noise. The abstract reads, individuals with identical genotypes exhibit great phenotypic variability, known as biological noise, which has broad implications. So this is where we have individual genotypes. So we might have wild types of, from different individuals, maybe 10 wild type genotypes, and there's variation in those wild type genotypes, but it's sort of random, and it doesn't really, there's no, strong mutation or deviation from that wild type. You do have defined mutants in C. elegans, for example, and in Drosophila, that are backcrossed with respect to a specific functional mutation. And so that's really the difference between the wild type and the mutant genotypes. But even within identical genotypes, or so-called identical genotypes, there exhibits great phenotypic variability. And there have been studies on this in C. elegans that the N2 wild type has a lot of variation across, like you get it from all over the world. It's the N2 genotype is supposed to be from Hawaii, but you can, even those are going to have a lot of variation. So this is all known as biological noise, and people study biological noise, and there's, of course, intrinsic biological noise and extrinsic biological noise. The intrinsic noise is the noise that you have sort of due to variation, like what we're talking about here. Extrinsic noise is the noise introduced by the environment, and so you can have a lot of both in a data set. But what we're going to talk about here is intrinsic biological noise. While molecular level noise has been extensively studied, in-depth analysis of cellular level noise is challenging. So this is where we're talking about molecular noise. This is the intrinsic noise and the extrinsic noise. What they're talking about here also is cellular level noise. So this is a little bit different. This is talking about this noise in cellular position. And again, it can be driven by noise in the developmental program, as it were, which is intrinsic, or it can be driven by the environment, which is extrinsic. So this is kind of what we're getting at. But we're talking about the cellular level, which is sort of manifest in something like a cell position or a cell phenotype. Here we present a systems level quantitative and functional analysis of noise in cellular position during embryogenesis, an important phenotype indicating differentiation and morphogenesis. So this is noise in cellular position is really indicative of differentiation and morphogenesis. We show that cellular position noise is deterministic, stringently regulated by intrinsic and extrinsic mechanisms. So cellular position noise actually is regulated by extrinsic mechanisms, but also intrinsic mechanisms. The noise level is determined by the cell lineage identity and is coupled to developmental properties including embryonic localization, cell contact, and left-right symmetry. So this is again where we have, position is governed by left-right symmetry, of course. It's governed by cell contact, so what are the other cells coming into contact with that cell? And it's general localization in the embryo. So remember earlier in the meeting, I talked about these point clouds. You have different observations of cells that can be within this point cloud. You end up with a centroid value, which is sort of the mean of that point cloud, the mean position. Now, that point cloud isn't infinite. It doesn't go across the entire embryo. It's going to be concentrated in a certain location in the embryo. Some of that is due to left-right symmetry. So we expect, say, a cell that's on the left-hand side to be on the left-hand side, and the right-hand side to be on the right-hand side, or an ABA to be anterior and ABL to be posterior. So there are some trends. They don't just occur randomly, but there's a lot of variation around those positions. Cell contacts, of course, make these harder. The cell can't move around infinitely as well. They contact other cells, so their migration is sort of constrained. So we can actually understand this variation in that context. And indeed, we shouldn't see just kind of a scatter of points. We should see maybe some patterns within those points as well. This is what I was talking about with respect to the hypernodes and some of the local sort of patterns within those hypernodes when we make that tripartite coupling. So we have like this mapping from the mother to each daughter cell. And we might have cells that are sort of in the left-hand side of that space, mapping to daughters in the left-hand side of that space. So that's what I meant by that. Cells follow a concordant high-low or low-high, low pattern of noise dynamics. So this is where you have low noise, high noise, and then low noise. And fate specification triggers a global downregulation of noise that provides a noise buffering strategy. So this is where when you get to a certain cell fate, there's a global downregulation of noise. So noise is actually present in a pluripotent cell or a totipotent cell at a higher level than what you would see in a differentiated cell. And the reason you have that is because those pluripotent cells, those totipotent cells, they have to maintain this stemness state, but they also have to sort of poise themselves for multiple transformations. So they can't be in a strong sort of ground state and then differentiate into a new cell type. And the whole point of pluripotency, you have a cell that's poised to maybe go in many different directions. And so when it finally finds its sort of identity, it downregulates everything. And in that way, it can maintain that identity in the face of, environmental pressures or whatever. So in C. elegans, we know that cells are deterministic with respect to their fate, but stem cells, pluripotent cells, and what they call founder cells in C. elegans maintain this higher level of noise. And this allows, the daughter cells to have different fates. So remember, we have mother cells, we have ancestral cells, we have daughter cells. The daughter cells from, say, like a founder cell like AB can maybe take on many different fates. And so the noise has to be at a higher level to poise the cell for these different changes. Cells follow concordant high-low pattern. So this is where you have this low noise, this high noise, and this low noise. So this is where we get this, we have low noise, high noise, low noise. Probably at the point of differentiation, you have this high noise, or at the point of fate specification, high noise, and then low noise after differentiation. Noise is stringently regulated throughout embryogenesis, especially during cell division and cell adhesion, and gap junctions function to restrict noise. So a lot of cells have gap junctions, and they're connected to one another through that. These are these electrical connections. You also have cell adhesion and cell division, so cells just can't float freely in the embryo. They have to sort of stay anchored to, a position with respect to the rest of the cells in the embryo. And then you have cell division, of course, which changes the complement of cells in an embryo. So these are all things that, play a role in regulating noise over the course of embryogenesis. Collectively, our study reveals system properties and regulatory mechanisms of cellular noise control during development. So this is a very detailed paper, and it kind of gets into some of the specifics of C. elegans. So in this paper, they actually have an approach to looking at comparing cell positions between embryos. So in this case, they've adopted a relative approach for representing a given target cell's position as a vector of geometrical distances from the centroid of its nucleus to those of all other coexisting cells. So this is this type of weighted average where you get these geometrical distances, and you use those as the weights to kind of determine that centroid value. So this is what I was talking about with respect to weighted averages. We then took CPN, which is this value as differences, and that vector between wild-type embryos for every cell at every aligned time point. So they talk about intrinsic regulation, cell lineage identity determines cellular position noise. Then they're asking the question using these CPN measurements, is CPN stochastic or deterministic? If deterministic, we predict CPN levels will be significantly associated with the developmental properties of cells. Vast knowledge available concerning C. elegans cells and embryogenesis allowed us to correlate CPN with diverse cell properties, including cell lineage, cell fate, 3D localization, and left-right symmetry. So they classified leaf cells, or the trace terminal cells at the 350 cell stage, into 12 groups based on the sublineages they derived from, and found that the levels of CPN were sublineage specific. So this is where you have the sublineage specificity. This is something we'll see with respect to, at the point of 350 cells, you have some differentiated cells, some not. But again, the founder cells are very general, and as you move down the lineage tree, the cells get more specific with respect to their ultimate fate. So you expect to see some changes that are sublineage specific. For example, cells from MS and P3 sublineages exhibited higher levels of CPN, while cells from the AB, PRA, and C sublineages exhibited lower levels. To further elaborate the relationship between CPN and cell lineage, we searched for CPN transition points on the cell lineage tree, defined as ancestral cells that give rise to two sublineages with significantly different levels of CPN. Any transition points were identified, suggesting CPN is strongly associated with lineage identity. As a bilateral animal, the C. elegans body plan is generally left-right symmetric. So we expect that these pairs exist on the left side and the right side, and they actually have similar behaviors because they derive from the same mother cell. We ask whether these left-right symmetric cell pairs exhibit similar CPN levels. Correlation analysis indicated that the CPN levels of corresponding left and right cells were highly correlated. Moreover, the divergency of CPN values between left-right symmetric cells was significantly lower than in mock cells. So this is where we observe this, connection between the left-right pairs in terms of function. And so this is something that we can expect because they're basically mirror images of one another. It's worth mentioning that this is not universal, that there are left-right asymmetries as well. So we can focus on left-right symmetries, which represent a majority of the lineage tree, but it's actually the asymmetric relationships that are more interesting. So this is where we get to temporal regulation and this low-high-low pattern of noise dynamics. So having established the influence of lineage localization on CPN level, we next sought to answer another question: how do CPN levels change over time? Quantification of global CPN at each embryogenetic time point revealed a smooth and dynamic temporal profile. As shown in 5a, which we'll get to in a minute, this profile follows a low-high-low pattern. That is, during early embryogenesis, global CPN progressively increases with time until mid-embryogenesis, approximately between the 64 and 128 cell stage, or I guess in the AB lineage, so that's 256, or from 120 to 256. Thereafter, the CPN level gradually reduces, and by the 350 cell stage, it reaches a level comparable to the beginning of embryogenesis. So this is noise, like what I was saying about noise, that noise, cells are poised to make changes, and what they're saying here is that very early in the lineage tree, noise is low, generally, and then it's at a sort of a high level as things are diverging and things are starting to differentiate. And then by the 350 cell stage, we get this lower level of CPN that's similar to the beginning of embryogenesis. And so, there are also linkages to global developmental patterns as well that we might be interested in correlating this with. We found the pattern of noise change to be tightly aligned to developmental stage rather than the absolute time of development. So this means that there's this developmental stage aspect that isn't that is sort of decoupled from time of development. So if you change the temperature in which C. elegans embryos are reared in, if you change it from like 21°C, which is near room temperature, to 16°C, you can actually change sort of the growth of the embryo or the timing of the embryo. So if I were to compare an embryo raised at 21°C with an embryo raised at 16°C, the timing would be stretched out, it would be delayed. And so there are going to be changes in noise that reflect this delayed timing at 16°C. So this is an interesting finding because there are some mutants that actually need to be reared at like 15 or 16°C. And they're viable if they're raised at that temperature. If they're raised at room temperature, they're not viable, which is an interesting thing in and of itself. But it does speak to this change of CPN, changing the pattern of CPN changes with respect to rearing temperature. And so, as I said before, fate specification triggers a global downregulation of position noise. We expect that position noise to be lowered as fate specification increases. So when we have changes of fate, they basically stop position noise. So those cells have to be in a certain position as they differentiate. So once these cells differentiate, they need to be in a specific position. In terms of CPN is under stringent developmental constraint. This is position noise being tightly controlled throughout embryogenesis, especially during cell division. So they want to test whether CPN is subject to tight constraint by comparing observed CPN levels to expected values based on the simulation. So they looked at the simulation and they found that the expected CPN was higher than the observed CPN for all cells, suggesting that in the cells it's more tightly regulated. So in conclusion, their findings shed light on the systems-wide regulation of position noise. First, we show that position noise is tightly controlled throughout embryogenesis, especially during cell division, to maintain a reproducible cellular configuration in embryos. So we should expect that because, of course, in C. elegans, we know that cells end up in a specific position, and there's very little variation across embryos, at least in theory, except for in the defined mutants where there may be specific changes. But even in the defined mutants, we know what those changes are. They're very apparent. And so this is interesting from this perspective of how do C. elegans embryos maintain this control over position. And this, of course, speaks to variation of position as well, because what they're looking at here is the variation of position with respect to the final outcome. And what we're talking about is something different, which are the different independent observations in that noise, but there are parallels between them because those cells need to be in a specific place. It is well known that in certain early blastomeres, such as EMS in C. elegans and ABAR, a mitotic spindle is tightly regulated during cell division to establish cell asymmetry or to place daughter cells at specific positions for signal receiving. Our data extend previous findings by showing the division of most cells is stringently regulated to place daughter cells at reproducible positions. So the daughter cells need to be in places, sort of take their places. And so we need to have this sort of control that has some noise, but it's also very tightly controlled. And that cell movements within a cycle are also subject to constraint, although to a lesser extent compared to cell division. Second, our data predict the existence of cell lineage-based intrinsic and cell location-based extrinsic mechanisms in determining position noise level. Together, our study provides insight into the systems, properties, and regulatory mechanisms guiding cellular noise during in vivo development. So I hope that was an interesting presentation on these two papers. Let me know if you have any questions.